organizadores del Congreso para invitarme. Es un honor participar con toda la gente de Latinoamérica. Realmente es un honor. Vengo de Vermont, el noreste de Estados Unidos. Es muy frío aquí. Saludos del menos 15 grados. Esperé estar en Buenos Aires, pero aquí vamos. Entonces, decidí dar la charla en inglés. Entonces vamos en inglés. Si tienen preguntas en español, está bien. Okay, so again, thank you very much for inviting me to participate in this conference. It's an honor. It's been an amazing conference, and I look forward to future conferences. It looks like this kind of a virtual conference is probably going to stay with us as a part of maybe normal conferences where everybody from all over the world can participate. Anyway, so I'm here to talk about species delimitation in some way. Maybe not the traditional way, but I'm going to talk about the dimensions of diversity in the Caribbean biodiversity hotspot, and basically how we in the CarBio project approach the collection and discovery and description of new species. So the question of this symposium is one or many species. That's the theme. My goal here is to approach this through kind of a practical approach to biodiversity discovery and species delimitation using massive taxon sampling, but maybe relatively few characters. I think there are a lot of ways in which we can inform on species discovery, species delimitation, in addition to maybe sophisticated methods, including lots of molecular data. We have lots of information from phylogeny, geology, distribution patterns, habitat, dispersal ability that informs us on both species identity and putative delimitation. I think we are currently in a biodiversity crisis, and we are working on a group of organisms that is highly diverse, but also vastly understudied, in the sense that probably what we know about spiders nowadays is less than 30 or 40% of the actual diversity. In other words, a reasonable estimate is that 70% of the diversity of arachnids is undiscovered, and maybe much more. So when we come to species delimitation, there are many highly sophisticated methods of doing species delimitation that require multiple, maybe hundreds of loci or genes to do sophisticated coalescence analysis and all that stuff. These methods are great, but they are a little costly and they take a little time. I think in the current era, when all of us are presented with limited resources, we have to think about how we allocate these resources. And I think at the moment, effective biodiversity discovery, we have to find a balance between the sophistication of our analysis and how much resources we allocate to getting tons of data for a few individuals. And on the other hand, our ability to go to the field and sample specimens and rapidly discover and describe biodiversity before it's lost. we are losing biodiversity at an alarming rate. We know that most of spiders and other arachnids remain undiscovered. So if I'm giving a resource, say a few $1 million, I think my priority, and maybe it should be our priority as a community, would be to invest these millions prominently in doing field work and discovering and collecting taxa in the field, and then using relatively simple and relatively cheap methods to discover and describe biodiversity before it's lost. And then subsequently, we have the specimens and we can allow ourselves to do more sophisticated analysis. That is my take anyway. So we have been running the Caribbean biogeographical project, or CarBio, for almost a decade now. And this project is fundamentally a biodiversity discovery project. We're trying to emphasize about 30 different lineages of arachnids and some non-arachnids as well. The fundamental goal is to set the stage for a multi-lineage comparative biogeographic project at a scale not hitherto tried. But we have gotten quite a lot of resources, but we have really allocated most of them to going to the field and sampling extensively. So the project has gone to over 100 collection sites. We have more than 200,000 specimens in our freezers, probably a lot more. We've sequenced about 20,000 specimens maybe. We've involved a lot of people, 5 graduate students and several part-time graduate students, lots of undergrads, postdocs. The whole project involves about 10 laboratories across the world. And to date, we've published about 20 papers on this project, and we've got a lot of things in preparation. So it's a big project. It's going on a decade. We're trying to get more money to continue. But the emphasis is securing specimens from all over the Caribbean primarily, and then subsequently to use those data to discover and describe species and to understand the biogeography of the region. So we went into this project with a theory in mind. So the whole project is grounded upon a theoretical basis, which is that we hypothesize that dispersal ability of taxa, the propensity to disperse, is very critical, not only in the distribution of taxa, but also in the diversity of taxa. So here's a very simple example. The theory that we have recapitulated, it's not ours originally, but we've represented it, suggests that taxa of intermediate dispersal ability should have the highest diversity in oceanic archipelagos. And here's a very simple example to demonstrate this principle from some nephilids in the Western Indian Ocean. We have Clitaedra, which is a habitat specialist. It builds its webs on bark of trees. It's not known to balloon. It's a very limited disperser. And this species, or this group, we can find on Africa and on Madagascar. These things used to be connected. So Clitaedra is presumably present in Madagascar vicariously, and it has not, because of its limited dispersal ability, reached any of the outlying Western Indian Ocean islands. So it has a limited distribution and limited diversity, therefore. We have nephila, which are excellent dispersers. they have been able to colonize all the islands, but they have such fragility that it looks like they maintain gene flow all the way from Africa to Rodriguez. This is a couple of 1,000 miles, and there's only a single species of Nephila in the entire region. Whereas the intermediate dispersal, Nephilingis, in this case, has colonized many, including Green Mauritius, but not all of the islands, but it's sufficiently dispersal limited to have subsequently speciated on some of these isolated landmasses, and the intermediate disperser is the most diverse in this region. This is a very simplified example, but it serves to kind of exemplify the point. So our hypothesis is that dispersal ability positively correlates with the number of islands colonized, which is kind of obvious, but the intermediate dispersers that are not constantly traveling between islands are the ones that have the highest diversity. So that's the kind of a conceptual basis for our project. So now I'm going to look at a few groups in the Caribbean, and we'll start with some taxa that we a priori suspect are really good dispersers, such as Nephila or Trichonephila. Nephilids or Nephila are found everywhere. They're on all islands. They're have occupied any land masses that we know of in the Caribbean. And it's notable that while they're very widespread, as predicted by their dispersal ability, they appear to be maintaining gene flow among islands. So that, for example, we see here across multiple islands in the Caribbean, specimens share a single haplotype, so that they are coming from a common stock. They are exchanging genes within the Caribbean, probably every generation, so every few generations. They have very low total diversity in the area, but they're widely distributed. Now, we think maybe there may be a different species in the South America, east and west of the Andes and the Caribbean region, but this remains to be tested. But the take-home message is that Nephila is an excellent disperser. It's found everywhere, and it has low species diversity. So in general, high dispersal predicts broad distribution and low diversity. Another excellent disperser, Argoper. We looked at this, in this case, Argoper arcantata in the Caribbean. We see very much similar patterns as we saw in Nephila. We find that this species is widespread. There is some correspondence between the locality and the phylogeny, but there is evidence of gene flow among all the islands. So in general, in these excellent dispersers, we find limited correspondence or no correspondence between the phylogeny and historical geology, but we find some correspondence between modern geography and the phylogeny. In other words, the species reflect the current positions of the land masses, and there's some subdivision of specimens according to the islands they currently occur on, but the distances are very small, and we see evidence of gene flow among all of these. Again, like in Nephila, most of the Caribbean islands seem to be sharing a population And we find some evidence for maybe additional species in part of the Caribbean. But the fundamental take-up point, a lineage with high dispersal has a broad distribution and relatively low diversity in the region. Yet another example, Gasteracantha, cangriformis. This is the only species of Gasteracantha in the New World. It's known to be an excellent disperser. As you can see, the Caribbean islands, there is some resemblance between the current geographical situation and phylogeny. There is no correspondence between historical geology and phylogeny. It appears that most of this group corresponds to a single species with ongoing gene flow among Caribbean islands. There is some evidence in our data that there may be a different species occupying the southeastern USA, but this is something that we would require sort of a higher level data to really test. But again, the take-home message, we have a taxon with high dispersal, it's found broadly distributed, and it has low diversity in the region. So it's very hard to document species with very limited dispersal because they're simply absent. You know, we have multiple lineages of very poor dispersers that are simply not present in the Caribbean. So how do we study them? We don't see them in the Caribbean. So this is akin to, for example, Clitetra in the Indian Ocean that doesn't colonize the outer islands, or for example, Lephistius that has been studied in very great detail recently, that are found on the Eastern Asian land and onto islands that were connected to the mainland, but not in the islands that have never been connected to mainland. So there's a tons of dispersal limited taxa that don't reach the Caribbean, therefore have zero diversity in the Caribbean, but it's kind of hard to really study them hard because they're not there. So most of taxa that we study are somewhere in between those extremes. They are not excellent dispersers, but they're good enough dispersers to have colonized the islands. And I'll take a few examples of those kind of dispersers in the Caribbean. One of my favorite examples is Denopis, the ogre-faced spiders. So we have a global collection of these guys, and the phylogeny of these guys supports a Gondwanan distribution. So over 100 million years ago, they were present on Gondwana before, for example, the New World and Old World split apart. And we see detailed correspondence between phylogeny and historical geology. And these species, these groups are quite diverse in the Caribbean because they follow the kind of a geological evolution of the Caribbean, which goes millions and millions of years back. So in the case of these kind of lineages, we see that phylogeny recapitulates geology. It doesn't necessarily reflect current geography, and we see relatively high species richness in these kind of groups. So a very similar example, here is Loxosceles. These guys are somewhat intermediate in their dispersal ability. Again, like in Denopis, we see strong support for a Gondwanan distribution, where the history of this lineage goes back over 100 million years, and they were probably shared before the continents separated. At the level of Gondwana, all the way to the details of the Caribbean geology, we see a close correspondence between phylogeny and geology. And
Sorry, you have 3 minutes to finish.
Okay, and we see a great diversity of these lineages. So when you have groups of limited dispersal, phylogeny recapitulates geology, and we have high species richness, and often we see closer match between molecular data and geography than between morphology and geography. So I'm just going to cut through this. It's just saying the same things, basically. When we have this kind of comparative data of multiple lineages that share the same arena and have similar dispersal biologies, we start to reveal biogeographic patterns that may show us underlying factors in speciation processes, so we can start to see some general things that may contribute to why certain lineages are diverse and certain other lineages are not diverse in the Caribbean. We have another example of an intermediate disperser, Spintharis, which shows very much similar patterns. We have evidence of a single colonization long time ago, probably via a land bridge, and a detailed correspondence between phylogeny and historical geology. Okay, so yet again, another example of this would be Phrynis, the amblypygids in the Caribbean. Before we started our work, it was thought that Caribbean amblypygid represented maybe 3 species across the entire region. What we have found, in contrary, is that we find strong genetic evidence of distinction between not only geological areas such as limestone and genus rock, but also we find that these species inhabit caves all over the place, and each cave and cave system also form unique genetic clades or clusters. So that when the question is one or many species, the answer often depends on the dispersal ability and the biology of the taxon, especially when you have taxa of intermediate dispersal ability, you tend to have higher species richness. And ultimately, the number of isolated habitat that a lineage occupies is a close predictor of the number of species that we find. For example, these guys occupy caves. They are unique genetically in almost each and every cave we visit. So they have a high, super high diversity because they are found in lots of isolated habitats. We now have tons of more data, 800 sequence specimens across the Caribbean. Again, we're finding this pattern where almost every place we visit, especially if it's within a cave, these guys are genetically isolated, unique, and potentially a different species. Basically, going broadly, sampling broadly, we are finding incredible diversity. The take-home message here is that when dispersal is limited and the number of isolated habitats is high, we may find exceptional diversity. I don't want to haphazard a guess, but
I'm sorry, Ingi, but we don't have more time. So okay. Thank you so much. I'm sorry, but we have to present.
That's fine.
Thank you so much. Amazing work.